אנחנו בשיעור ראשון
כן. Big Shahada. Without the warm-up.
אתה מלמד או שאתה עושה גם וגם? עליי כן. לא, עליי. Actually, I'm not going to be the presenter today. I'm going to be Eden. But we'll give a short intro of this class, and then we're going to start. Actually, I'm going to give a short intro, and then we're going to do a quick quiz, and then we're going to start. So we are in the course of human neuroscience methods, which is in a way should continue or add on the method in neuroscience that exists in LSEC. And in here, we're going to try to well, first I'm going to introduce myself. My name is Aviv. I'm in LSEC for the last 3.5 years, I think. I'm sitting here, and if you need me, you're welcome to come and talk with me or with Shai about anything that is related to the course. I didn't say, but Shai, she's the TA of this course. She will, it's more than TA. She's actually gonna work with you in three. There will be 3 meetings with Shai that will be kind of lab, hands-on, working on the topics of the course. So that gets me to the topics of the course. And what we want is for you to have some knowledge about research in neuroscience in humans. And I'm not expecting that after this course you will be ready to do research in neuroscience, but at least when you're going to read an article, you will be able to read it and kind of like say, okay, I understand what they did, I trust what they did, I don't. And this course was done last year with Leon Duell, and he will come back next year. In this semester, he's in sabbatical, and it's covered MRI and EEG. This is the main 2 topics that we cover. And last year, the lab was separated from the course, and it ended up being a very hard course because there was a lot of theories and like, I don't know, the physics of and whatever of the measurement. And most people didn't took the labs because that was it was an optional. And so we decided to make the lab non-optional and remove some of the material. So this course is more condensed. You can see that we're covering EEG, MRI, which is around MRI, the machine, and then MRI, how we measure brain anatomy, and then fMRI. Basically, this is it. We're covering really core methods that widely used in human neuroscience. There are many other methods which we cover in the other course, which we don't this year. And we're doing that with the hope that at least what we cover you will actually understand, and there will be labs to practice it. Okay, so that's the hope. And so the first 2 classes will be Eden will talk about EEG. Then I will cover MRI first, the machine, what it does, where the signal is coming from, and then we're going to talk a little bit about brain anatomy in general, and then particular brain on methods for diffusion MRI, which is a very important tool in brain anatomy. Then I will discuss there will be a lab for EEG, there will be a lab for MRI, which in there you will actually play with data or we'll simulate data, and then we try to analyze it, see what is the protocol of analysis. At least you will have a sense of that. The next part will be about fMRI. I will first cover the biophysical source of the signal. So it's the neurovascular coupling that derives the fMRI signal. And then the next two classes will be with Yuval, which is the head of the ENU, the imaging unit of LSEC, which is expert in fMRI. And he will tell you a little bit about how we analyze data measuring function in the human brain. And then we have another lab for fMRI. And the last class will be me discussing what I see as the future, or the things that's like really currently happening in probably will be in MRI and fMRI, but kind of like well, we'll discuss where this field is going or heading to. What else? Labs. We hope to do the labs here. But that means that you would have to use your own computers. The other option is that all the labs will be in a computer room, which is, do you remember where it is?
Where it is.
So there will be a discussion between you and Shai.
Does anybody not have a PC? You don't? All right. You have your laptop?
So essentially a laptop. Doesn't have to be a PC.
Do you have MATLAB on your laptop? Yeah?
Laptop
If
exists.
someone doesn't, please email me or contact me.
That will make a difference for us, if you have it or not. So that will change the room, essentially. The aim of this class. So with that, I just copied kind of what we said in the syllabus, and I kind of mentioned that already. What we want you is not to be perfect in just go tomorrow to the lab and start doing research, but actually I think it's even more important for the people who does not planning to go to do research with humans. So assuming you're gonna work about all your work is gonna be theoretical work, or maybe it will gonna be some system neuroscience in mice, and you would notice that sometimes you're reading articles, and that kind of give you some kind of thoughts about your own work, and kind of like give you where the field is and so on. And most of the time you will read it and say, maybe I don't know what they measure, and I don't want to have that. I want you to be able to say, I understand what they measure, I trust what they did, I have a question about this specific. So that's what we really want in this course, for you to be able to understand the method in some level, so you can be a critical reader, for instance, of an article. So that's essentially what I just said. Besides also being able to at least know basic tools, and that's what we're going to do in the labs. So assuming, and just for example, you're really interested in LSD, and you want to understand what this article about multimodal imaging, neuroimaging, and you see these graphs, and they did resting state fMRI and fMRI, and then they did some electrical recording, this case with MEG, and they put everything on the cortex, and you look at that, maybe I understand what is the difference here, and I hope that, for instance, after the next two classes in EEG, you will be able to look at this graph and feel comfortable with that. You will have some sense of what was going on in here, and so on with the other things, with the other modalities. Okay, so what we expect from you. You already, Shai sent you these messages. We're going to try this year, the first time. Last year it was really hard for the students to arrive to the class, and there was so much material that we were like describing in the slide, and it was a little bit overwhelming. So to try to reduce this overwhelming experience of the classes, we want you to read something, and we try to find or either read or watch. When we can, we actually provide videos, which I think is easier, maybe easier. You tell me. I would be happy to get input if these materials are actually helpful. So the idea that you will come to the class and you won't be like totally naive about this topic. You heard about it already, and just to make sure that so this is some work, right? You need to spend an hour or two at home reading or watching, And so to get credit for that, there will be a quiz and the grade, and the quiz will be hopefully very easy. It's supposed to be. We try to make it easy, just kind of like, have you read? You kind of understand the big picture, and if so, you're gonna get. This quiz will actually add on to the final score in the course, and that's really in order to initiate a real discussion in class. So you would know something, you read about it already, and when the teacher teaches, you can actually be part of the discussion. There's not everything just above your head, and so the grade in this course is it's about 30% the quizzes. There are assignments in the labs. So in each lab there is assignment. There is no quizzes for the labs, but you expect to come with, let's say, already downloaded data and put whatever the toolbox that are needed in order to do the analysis. So the class, so both of those things will be counting as quiz. Either be ready to let the lab or be ready to the class. Okay? That will be testing the quizzes. There will be the assignment in the labs, which you will learn something. You will start some assignment during the lab, and then you will continue at home. And after a few weeks, and Shai will decide, but we're not going to pressure you in that. But after a few weeks, you will have to send the results, okay, of your analysis. And then there is being part in this class. It's also part of your evaluation, the lab and the classes. And then the last thing is that in the there is a day within the exams period, I think toward the end, in which we're going to do presentation. We depend how many will end up staying in the class. Either be each of you, or maybe we'll do it in couples, or maybe a bigger groups that will either present a topic. That's one option. Hi, you went to the labs. You're looking for you. I'm sorry for that. So it will be either presenting a topic which we didn't cover. So there were several topics we didn't cover, and then you will, in the presentation, you will cover it, and that will be done. I will give more instruction. That will be done relating the new topic, let's say TMS, to the topics we already learned. So kind of like relating it to why it's better, why it's different. Or the other option, if you have your own data, or you want to have some data and you want to do some analysis, or if you want to do some kind of small research, which I think is harder, but we also, I'm promising when we test or when we give a grade for that, we'll take it into consideration. You can also do a small kind of work with data in order to and present it in this last in the last class. So whatever you would prefer. In both cases, you would have to give some background. So that's part of this final project work and presentation. Okay. I think this is it. Any question before we actually go for the real thing? Which is Eden? No. Okay. Yes? No. So we're going to do the quiz, and then 5 minutes. We're going to do it very short. It's true, false. 3 questions of true and false, right? וואלה, אני תכף אציג אותך. לא עובד? בדקת?
שי, זה עד 3:30?
כן, אבל זה יכול להיות עד... לא, זה לא 3:30, זה בישיבת רם. לשלוש רבעי שעה, רבע שעה,
רבע שעה?
אני אגיד לך,
כאילו יש עד...
אני יכולה להגיד לך בגלל...
ברבע יש הפסקה של רבע שעה ועד 3:30. אבל יש לי חצי שעה עכשיו.
לסמן לך עכשיו?
לא, אני אצא.
Can we start? Okay. So I want to introduce Eden, which is first thank you because he didn't have to do it. He just agreed to do it. Then he's abroad and he's like our as good as having Leon here presenting about EEG. Eden is like as senior as you can be as in LSEC PhD student, right? Just about to finish his PhD, has multiple work dealing with EEG. Thank you so much.
Yeah, hi. So I'm very happy to be here. Actually, it somehow happened that it's the first opportunity that I have teaching an LSEC course, or a lesson in LSEC course. So I'm really happy to have a chance to do it. Really, the last moment before I finish my PhD. So yeah, my name is Eden. I do my PhD in Leon de Wells lab in the psychology department. We do mostly EEG, so a little bit of fMRI too. So we have just two classes to get through everything that at least some of you, this will be the only opportunity for you to deal with EEG or even human electrophysiology at all. So we have a lot to get through. And today what I want to try to go over is, first of all, a general introduction to the EEG signal, where it comes from, what we're measuring, a short discussion of the event-related potential analysis, which is the very basic approach to analyzing EEG as data. Talk about some famous ERP components, and we will understand exactly what this is later, and basically to show them as examples of how we use EEG for mostly neurocognitive research. And if we have time, I'll give you a quick introduction about spectral analysis in EEG. I think if we won't have time, then we do it next time, because I think it's pretty important. All right, so let's dive into the EEG signal. EEG was invented in the 1920s by Hans Berger in Germany. This is just an example of one of his recordings. You can see an EKG signal from the heart below, and this is the EEG signal recorded from the scalp. Actually, the first thing that he saw, which is also the first thing that we usually see when we look at EEG, apart from just noise, is something called alpha oscillations, which is these bursts of very clear rhythm. It's about 10 hertz. And even back in the 1920s, from about the 1930s, it was discovered that they have something to do with attention. And if we're going to have time to talk about spectral analysis in today's lesson, then we're going to go into more detail about what we're doing today with this kind of research. But this is just to show you where it started. Yeah, so So what is EEG? EEG is the electrical signal we measure on the scalp. EEG comes from electroencephalogram. Encephalos is head in Greek. The signal we're measuring has a very high temporal resolution, because whatever is happening in the electrical activity on the brain is measured in real time, manifests itself in real time on the scalp, and we can measure it. So we're talking about millisecond resolution, or sometimes even higher. What we are measuring is local field potentials. We'll get more into this, what this means in a bit. And we're also recording activity that is summed up over large parts of the brain. EEG has a very coarse spatial resolution, meaning that when we put an electrode on the head and measure what's the electrical activity on that part of the scalp, we're actually, this activity is influenced by what is happening over a very large part of the brain. Actually, you can say even all of the brain. So it's very hard to really localize the source of activity spatially, and we'll also get into that soon. Okay, what we're actually measuring is electrical potential. Electrical potential is basically a differential measurement, meaning that we're measuring a difference between two electrodes. Okay, it's just like measuring height. There's no absolute measurement of height. You have to compare it to some kind of baseline, sea level or whatever. So in EEG, we always have this baseline measurement. Physically, we just use a different electrode as our baseline, and this we call it a reference. This baseline. The choice of reference really varies. Sometimes you can use a specific electrode, like another electrode on the head or on the nose or behind the ears. Sometimes you can use what I usually use is the average of all of the electrodes. Of course, physically, when you measure it at first, it has to be compared to a specific electrode, because it's a physical measurement. So it has to be compared to another physical measurement. But then offline, you can change the reference to whatever you want. So you can change it, for example, to the average of all of the electrodes. So we're just going to give a recommendation that whoever is interested in exactly where the signal is coming from, this is a very good review. Actually, it should have been, I think, one of the next few slides when I actually talk about this topic. Okay. So let's place EEG in terms of its place in the spatial and temporal dimensions relative to other imaging methods. So this is EEG. It has a very high temporal resolution, like we said, and a very low spatial resolution. fMRI, for example, would be here. It has a better spatial resolution, but a worse temporal resolution. Yeah, I'm not going to get into this too much. I just want to add that since, I guess, this image came out, there's another another method of imaging in humans which becomes more and more prominent, which is called ECOG, or the electrocorticogram. This is basically, instead of having electrodes on the scalp, on the encephalos, it's having electrodes on the surface of the cortex. And in humans, of course, this is done only for clinical reasons, specifically for the surgical treatment of epilepsy. So people who have severe epilepsy that doesn't respond sufficiently well to medication usually have to go a surgical intervention where the epileptic focus has to be surgically removed. And to do that, there's a initial stage where they undergo surgery where electrodes are implanted on the surface of the cortex, and then they're monitored in the hospital for a couple of weeks, and seizures are recorded, and this is how the focus of the epileptic seizure is determined. So in many places, many hospitals where this kind of cooperation exists, you can go up to these patients and ask them to, while they're sitting in the hospital with electrodes inside their brain, to do a couple of experiments. And this is really, so it's a more complicated way to collect data logistically, because you're working with patients and in hospitals and you don't have a lot of time and you're not in a lab, on one hand. On the other hand, the quality of the signal itself is pretty amazing. I had the fortune to work with this kind of data, and it's really incredible if you're used to EEG and measuring from the scalp, measuring from the brain in the same very fine temporal resolution, but having a sort of, you can say more or less fMRI scale spatial resolution is really incredible. Yeah, so this is just a few words about ECOG, but we're not going to talk much about this. Okay, I didn't know there's sound to these videos. So okay, yeah, this presentation is, I didn't create all of it, so there's some surprises in it, I guess. Okay, so this is a video from our lab, an old video from our lab, showing just the basic process of how we work with EEG, how EEG is measured. So I'm going to go over this quickly. Basically, EEG is measured with this kind of a skull cap you put over your head. For this purpose, the head is first measured to find the correct cap size. Apart from the head electrodes, we also use a few extra electrodes, like you saw here, placed behind the ears. This is, for example, good for as a reference, or as a good way to pick up muscle noise, to use it later to cancel it out. This is then putting the cap on the head. Just gonna jump a bit ahead. All right, and then a few measures are taken to really fix it into place. Sorry. In here. Oh, okay. The standard method of placing electrodes on the head in EEG is called the 10-20 system. Basically, all this means is that if you take the head and you measure from the inion, which is the bump that you have on the back of your skull, just where your neck, really on the top of your neck, all the way to the nasion, which is what's the opposite of bump? The part just above your nose. So you take this distance, and then you put the first electrode 10% of that distance, and then the next one 20%, and so forth. So this is the 10-20 system. It's just a very standard way of making sure that we're all on the same page of where each electrode really is. So the next step would be to connect the electrodes, and this is done by injecting electrolytic gel, which will pretty much connect the scalp, the skin of the scalp, to the electrode. So it's just a conducting substance, because we're not trying. Obviously, we're not going to push the electrodes into the skin, and if they're just going to rest on top of the skin, especially since most people have hair, there's not going to be good electrical contact. So this is why we use this kind of gel. So this takes a while. You have to chat with the subject. So that's another important skill you need to have. A few more electrodes are usually placed around the eyes. This is a very good way of recording eye movements, especially if you don't have, if you're not using eye tracking, then you can use the signal recorded around the eyes, because each eye movement also changes the electric field produced by the eye. A nose electrode is usually used for either reference or as a way to identify muscle noise. Yeah.
So what's the naming rule for each electrode on the cap?
So yeah, so they have standard names. So I think you could see, yeah. So you know, FP1, F3, F7. Basically, this comes from, so this would be occipital, parietal, central, frontal, I guess, and then the number is just from side to side. Yeah, don't ask me why FP or why what's the difference between F and P and FP. You just get used to these names. Yeah, and by the way, any questions that you have, feel free to stop me. I'm trying to go over a lot of material, so whatever is not clear or you want to ask, then just stop me. Right. Okay, and then the next thing that we do is we just look at the signal. So this is pretty much what you see when you connect your subject to the recording machine, and you see this signal on the monitor. And again, like I briefly mentioned before, the most prominent neural signal that you're going to see, actually the only clear neurological signals you're going to see is alpha oscillation. So I think it's easier to see them. Yeah, so this, for example, this kind of oscillation. So this happens, as you can see in the video here, this usually happens when the subject closes their eyes, and this is because alpha power is really associated with allocation of sensory resources. So when you're attentive to your sensory input to the outside world, then alpha power is usually attenuated, and when you're more, let's say, oriented inwards, especially when you close your eyes, then there's an increase in alpha power. So it's very easy to see these bursts of alpha oscillations when the subject closes his eyes. Of course, it's not shown here for some reason, but the easiest thing that you can see in EEG is muscle noise. So, for example, if here, instead of closing his eyes, he do something like move his nose or tighten his jaw or whatever, then you'd see this really loud noise, which is really far stronger than anything you can actually measure from the brain. So it's also very important to ask subjects to remain calm, not to to have any tension in their face muscles, not to move too much. Yeah, it's really sensitive to that. And of course, you can try to clean the data later, and there's some methods to do that, but of course you want your measurement to be as clean as possible to begin with. Okay, this is just a very quick run-through of what an EEG experiment looks like, but let's talk about what we're actually measuring. So let's think about a single neuron, and what happens when the neuron receives input from another one. So basically, we have excitatory synaptic input from another neuron into the soma, or into and this input depolarizes the membrane, meaning that it becomes more negatively charged inside the cell. But outside the cells, it means it becomes more positively charged. Am I confusing? Is it the other way? Sorry? Yeah. So in this case, the soma would become. I'm not sure why this is. Yeah. Yeah, right. Yeah, so the outside of the cell becomes more negative. Yeah, okay. Yeah, so I was saying the other way around. Okay. Yeah. So anyway, so the part of the cell around the soma becomes negative, while the far side of the cell is still positive. Okay, so basically this creates something called a dipole. Okay, so a difference in the electrical potential between two spatial sources, spatial places, and this creates an electrical field. Okay, so basically current would travel around in this kind of pattern. Okay, so like this, and like this, and like this, etc. Right? So now we have something. So dipole is a very abstract physical entity which has 2 points with a difference in electrical potential between them. And when we have this kind of biological structure, of course, it's much more complex. But we can call this, you can use the term equivalent dipole to say this is more or less can be represented, can be simplified as this kind of dipole. And you can go from this to actually a group of neurons that are oriented in the same way and say all these neurons actually act as one dipole on a certain larger scale. So when we measure, so when we put, so if this electrode is oriented, this neuron is oriented in this way relative to the scalp, so this would be the scalp, and we have an electrode over here, then what is happening is that this dipole would create an electrical field such that we would measure a difference in potential between this part of the field and this part of the field. Okay, so a different way of saying this is that this would be an electrical source, meaning that current flows away from this point, and this would be an electrical sink. And so when you put electrodes here and here, there is a difference in potential between them. So there is some kind of voltage here. Is this clear? I'm really hand waving. I'm trying to, this is not a physics class. So I'm really trying to make this as simple as possible. All right. So if this would be our reference electrode, and this would be one of our head electrodes, then the signal we are measuring is actually this difference in potential caused by this dipole source. Okay? Yeah, so if we have this input into the soma of the cell, and of course it happens only for a short amount of time until the depolarization goes through the entire cell. So if we measure here, we have this temporary increase in this difference of potential. Okay, so I mentioned something before about many neurons being oriented in the same way to act as a certain dipole, and this is very important because let's say we have two of these neurons side by side, then if they are oriented in the same way, meaning that the cell body is located is here and the axon is located here, and excitatory input comes into here, creating the same kind of dipole, then you can just simplify this and say this is just one dipole, okay? And of course, this could be thousands of neurons, and not just two. But if these neurons are oriented in opposite ways, then this would be what's called a closed electrical field, meaning that the current just goes this way, and there's no electrical field created outside. So in this case, if you put an electrode here, you won't be able to measure anything that's happening here. Okay, so this is a closed electrical field, and it doesn't affect your faraway electrode. Meaning that so what we take away from this is that we can only measure changes in the electrical field if their source is sufficiently aligned in relation to our electrode. Okay. The place in the brain where you have a lot of neurons aligned in the same way is, of course, the cortex. Okay, and this is the reason why most of what we're measuring, or the signal that we're looking at in EEG, can be assumed to come from cortical activity. At the same time, the cortex is also highly folded, as you know. So there can be cases where if we have two sources on opposite sides of a sulcus which are oriented exactly one opposite another, they could, in theory, cancel each other out. And we'll see an example of something like this maybe a bit later. The second thing that's really highly important about measuring EEG activity is synchronization. So let's say we have these two electrodes which are oriented in the same way, in the same direction, but or let's say we don't have two, but we have a few thousands or a few millions, and each one of them receives this synaptic inputs, but not at the same time. Okay, so each one of them receives it at a different time. Then when we aggregate over this entire, over the electrical activity produced by this activity, then we're not going to see anything changing, right? Because on average across this entire population, nothing is really changing. So we're going to be able to see this kind of measurable change in the recorded potential on the scalp only if enough of these changes happen at the same time with the same orientation and the same, of course, electrical polarity. So if we have a positive and negative fields happening at the same time, they're going to cancel each other out. So everything has to be synchronized enough for us to be able to record this on the scalp. Correct? Yeah.
Question?
Yeah.
Doesn't this really interrupt the way you draw your conclusion from the measurements? If you don't know what you lost in the way.
Yeah. So it's a good question, but the thing is that what you lost on the way is pretty much almost all of the neural activity in the brain, because so you have all of this, hundreds of billions of neurons working at the same time in the brain, and you have like these 70 electrodes on the scalp measuring a highly correlated signal. So of course you're having this very you're losing almost all of the information, right? You're measuring something. Yeah. So actually the problem I think that you're actually talking about is that we don't know what exactly is the source of the activity that we're measuring. Okay, it's not like in multi-unit activity or even in fMRI where you can say, okay, we measured this signal and this activity that we measured is coming from this specific place in the brain, right? Because first of all, you don't know exactly where it's coming from. Second of all, you don't know if what you measured is, let me put it this way. When you do EEG, you don't assume that what you measured on the scalp is actually, so if you measure this kind of response, kind of waveform on the scalp, you never assume that there is a specific place in the brain which produced this specific response. Okay, because you always have to assume that whatever you're recording on the scalp comes from summing over activity over large parts of the brain. Some of this activity was picked up, some of it was cancelled out, some of it never reached the scalp because it was didn't have enough power, because it was attenuated too much on the way across the scalp. So you never assume that whatever you're measuring came from, uninterrupted from a specific place. But it doesn't mean that you can't. And we're going to talk about later about what kind of inferences you can make. For example, if you're comparing two conditions and you see that this condition created that these conditions differed in the EEG response that you see around 100 milliseconds after the stimulus, then you have no problem of inferring that the brain processed these conditions differently at around 100 milliseconds. Right? It doesn't matter where it came from and what you lost on the way. All right. Let's talk for a second about source depth. So it really matters where the so this dipole we talked about, where it's located inside the head. So here, for example, you have these two examples of cortical dipoles, or not cortical actually. This is not cortical. So brain dipole. So a dipole located near the surface of the brain would create. Surface of the brain would create an electrical field which drops off very quickly. So if you start recording here, and you have an electrode here, 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 and here, we'd have this very quick drop of potential, right? Because this would be very positive, and this already would be negative, okay? On the other hand, if your dipole is here, then you have a very different shape of electrical field, where the gradient of how fast the potential drops from here to here to here is much slower, okay? And only here it's negative. So different dipoles create different topographies on the scalp, and this also means that you sometimes have to be very careful about that you can never make naive, or you should be very careful about making naive interpretations of seeing something, of translating from the scalp to the brain. So it's usually convenient to say, okay, I see this waveform that is positive in the back of the brain, so probably it comes from an occipital source. And then I see another one which is frontal, maybe it comes from a frontal source. But it's not always the case. And sometimes you can have two dipoles, two sources, which are very near each other inside the head, but because they are oriented in different ways, one of them would create a very localized topography here, and the second one would create a different topography here. And you maybe would tempted to say, okay, I found these two sources of activity here and here, on the scalp here and here, which seem to be very correlated, so maybe this kind of long distance correlation between these two areas in the brain, where actually there's no long distance correlation, they're actually very near each other inside the head. So you have to be very, very careful with these inferences. And I think it's one last point about this signal is that since we're talking about electrical fields, and up to this point I've been kind of talking like the head is this kind of a perfect sphere that doesn't affect the signal, but it's not. It's biological tissue, and with each kind of tissue having different conductive properties. So this kind of abstraction where you have this dipole and this is the shape of the electrical field is not right, because the electrical field is affected and distorted by whatever medium it passes on the way, especially the skull, which is not very conductive to electrical current. So it makes it even harder to translate from whatever you're measuring on the scalp to where it came from in the brain, because there's this complicated model of how exactly this kind of dipole would be expressed on the head once it passes through all these different tissues. So of course we would, okay. Wait, so we have to go now, right?
I mean, usually we break at the quarter to and come back at 3.
Yeah, okay.
We have like 1 hour.
Yeah, no, we can take a break now. Okay.
אחד מהסליידים הראשונים, מי שראה את למה הדיפול הוא דרך כלל בכיוון הזה, כאילו גם פה וגם פה יש פה אותם פלוסים כאילו הדיפול אמרת שזה דרך כלל בכיוון כאילו של הדיפול של הנוירון, נכון? כאילו מהפלוס הזה למינוס
הכיוון של הדיפול זה
לא, אני מתכוון כאילו לא לכיוון של האפקט, אלא לכיוון של המטריץ, כאילו של כן, בדיוק.
למה זה כזה ולא כזה?
כן, מה ששינה זה רק אמרנו שפה יש איזשהו זרם חיובי שנכנס, נכון? ולכן המתח כאילו פה יותר יותר חיובי.
אבל אם אתה מסתכל, שם את כאן וכאן, אז לא יהיה ביניהם מתח. הם אותו פוטנציאל.
לא, נכון, אבל גם בין פה לפה כן יש.
בין פה פה כאן.
כאילו בין כל נקודה, חוץ מהנקודה. בין נקודה כלשהי לנקודה הזאת, אז כן יהיה איזשהו מתח.
כן, בנקודה כלשהי, כי זה היה בדיוק
כן. אז למה אתה אומר שהדיפול דווקא באוריינטציה הזאת ולא כאילו באוריינטציה הזאת או באוריינטציה אחרת?
דווקא הוא כזה, כן? ככל אם יש לך, שוב, פוטנציאל זה מודד תמיד בין שתי נקודות, צריך לבחור איפה זה אותם שתי הנקודות. יכול לבחור שאחת נמצאת כאן, אבל זו החלטה שרירותית. אבל בפועל זה כאילו הסינק והסורס נמצאים כאן, ועכשיו יש הסורסים שתי הנקודות.
מה שאמרתי זה למדת דווקא איך קראת הסינק והסורס, מה זה הסורס, זה הסורס וזה הסינק?
אני לא חודק זה הפוך.
לא משנה, אני מבין איפה אתה, כאילו המינוס הבנתי, כאילו פיזית קורה פה משהו, כאילו...
לא, המינוס זה רק ביחס לזה. זה שקורה פה משהו, זה שקורה... זה רק אומר שיחסית לרסטים פוטנציאל שיש פה, כרגע נהיה פה חזוקה שלי. עכשיו, ביחס לכאן.
נכון, וגם פה לא יש תהליך הזה, כאילו בכל מקום אחר במוח לא יש תהליך הזה.
אני לא ספציפית את הנוירון הזה עכשיו, בכל נוירון.
אז כאילו...
יש לי אינגש? הייתי מדעים, לא, כאילו, זה לא עובד.
לא, זה לא עוזר לי. כאילו, אני שלח, לא משנה. זה יכול להיות.
הנקודה היא שבסופו של דבר, נניח שזה היה נוירון היחיד שקורה פה משהו במוח, כל הנוירונים האחרים נמצאים כולם, כל אורך הנוירון זה 170 מיקרוולט, ורק פה פתאום החלק התחתון שלו נראה הרבה פולריזציה. אז עכשיו בנוירון הזה יש הבדל בין הנקודה הזאת לנקודה הזאת.
כאילו אתה מסתכל משמע נצחה זה שאתה מוקרב בתוך אותו נוירון.
לא חניין, זה גם יכול להיות שזה עכשיו כאילו, שזה הקורטקס, כן, זה כאילו הבדל בין שכבה ושכבה שיש.
היי, אני רציתי בכלל לשאול כמה משתנה מהסדנה של אימא, אתה יודע מה אני מדברת? לקחתי את הסדנה שאוהבת סדנה ב-12 א', ממש אני יכולה, זה היה יומיים. זה היה יותר, טוב אולי אני צריכה לדעת את המשלים שלך כדי לדעת אם זה לא קשה.
בטוח, אני לא יודע מה רוצה, בטוח
אני מניחה, קודם כל אני מניחה, אני אגיד לך מה, אני בסדנה של דעת מה שקרה.
בשני השיעורים שאני עושה, שי כן תעשה משהו. שני השיעורים שאני עושה, אני לא מדבר על הנהגים בכלל. נכנס לטיפה.
כן, אבל זה נגיד ממש כללי, נכון? כי אני כבר עובדת עם כל מה שאתה עד עכשיו, זה ברור לי. זה כאילו, אני מניחה שזה יהיה להם די מבוא.
אז הרוב זה מאוד מאוד בסיסי, בסוף אני מדבר קצת על אם אתה קצת להסתק בין
כן, אז זה לא ממש יועיל לי, כן.
שוב, אם את רואה את זה כאילו ממש שיעור הראשון.
נכון, זהו, אני מניחה שכן. לא טוב, בגלל זה באתי
השיעור השני אולי קצת, אני לא ברור לי גם על מה יהיה בשיעור השני, כנראה יהיה על הפרקטים,
זהו, זה אני בעד.
אולי קצת על איך לחלק משתנים בתוך איש, וגם הדבר הכי פרקטי אולי שדבר עליו זה בעצם עושה את הסטטיסטיקה
אוקיי, כן. אז כן. וזה יהיה השיעור השני בעצם. לא, זה ההתגרות שלי עם לרשם הפרוצציה MRI. כמה שהוא מעניין, לא זהו, נראה לי, כן, נראה לי זה לא יהיה פרוצציה ממש. נורא מבקש. כן, טוב. אולי אני ממשיך מהבאה לעשות משהו. תודה.
אני גם כאילו, אני גם כזה, מגמגם כזה, כל החלק הזה שזה כל כך שאני לא מתעסק בכלל בדברים האלה.
אני יודעת.
כאילו למדתי את זה, כן, כאילו אני רק רוצה לדבר, כאילו על איך לנתח,
חשבתי הרבה פחות.
זה גם, לא, זה לא יהיה, כאילו זה רלוונטי רק לאנשים שממש, טוב, ממש, זה יהיה עם שי.
אז לא ממש, נגיד הסמלה שלו מעולה. מהבחינה הזאת, זה ממש להמשיך ש... תודה.
רגע, איפה את עובדת?
אני במעבדת המלאך.
אוקיי, כי אצלנו לפחות לא יצא אחרי בישהו במעבדה.
לא, למה?
פספס, כי היה לנו נגיד פספס. אני עברתי לפני כמה...
וואי, באמת? מה עברת?
שתי ישיבות מעבדה שממש עברנו, כאילו אני בעיקר יצאתי סקריפטיה. זה הזדמנות לעצור כאילו בכל השלבים של הניתוח.
קודם כל אני אקח ממך את הניתוח, כאילו אולי אני אראה אותו. אני עובדת, מכיר את סגי, אז אני עובדת עם במחזור שלך? אז אני עובדת עם סגי גם, קצת חיובי כזה, זה פחות מובן, אבל זה סגי כזה. אז זהו, כאילו בעיקרון אני בסדר, פשוט אמרתי אם יש משהו מעבר, זה יהיה נחמד.
לא, זה הידע כזה שיותר במעבדות.
כן, כן. אני אדבר עם הרב שנעשה את זה. פעולה שהמעבדה רק ישמע אותי. טוב, תודה. שח. היי, נעים מאוד. אנחנו ממש מעבדה כאילו... זהו, נכון. כן? זהו, דווקא התגנבתי למעבדה שלכם מדי פעם ושאלתי דברים. כאילו הרבה דברים שנגיד לי לא הולכים, ואני גם מתעסקת עם אוטיזם, יותר בעייתי איתם. כל הניתוח. כאילו כל הזה של זה.
זה שמדברים על הראייה, אנחנו מדברים על השנייה, אבל זה אנחנו מדברים על הראייה.
אם כבר. אתה מתעסק עם ניתוח של מעט סגים?
אפשר להגיד, באיקון זה...
נכון. כן, אבל כן. לא יודעת איך להשוות עם זה, אבל... סתם כי פשוט יוצאים, כאילו למשל שאני מנסה לחשב את הטאו, למשל יצגבו הזמן של... אוקיי, אני מודדת את הפטציה, ואז כשאני מנסה לחשב את הזמן שלהם.
רגע, כל זמן שלהם.
כאילו של מתי האדפטציה יורדת. אני נותנת להם... אוקיי, אז אני חושבת את זה ממש להסביר, כן, זה... לא, הקטע זה שפשוט אני אקח נתונים והסטמפל שלי הוא לא מספיק גדול כדי לתת משהו שהוא באמת... כן. האם אתם משתמשים של סטטיסטיות? מדהימות.
אני עושה הרבה סטטיסטיקה, עושה לי הרבה סטטיסטיקה לא פליטקים.
אוקיי. זה בגלל שאתה עושה את זה.
כן, גם ב-EG נגיד ב-EG אני מסתכל על תקופות גמא.
נכון, שעובדים גם.
והעניין הוא שרואים את המוגסן של מעט מתקן. ואז אני מצאים שלך 15 מלכים, זה עושה לי 4 שיש בהם תגובה, אז זה לא אכפת לי.
אני במצב טוב איתי.
ואז אני פשוט... אין מה לעשות, כאילו, יש שיטות, אני חושב, שלא כך מכיר, שאומרות, שלעשות את secondary analysis, להגיד שיש לך 4 ניתקים, והגמרה שזה מספיק נדבק ב4 כדי... אבל אני לא עושה את זה הפעם, אני כאילו מראה, כאילו, 4 ניתקים, נתן לי אפקט נדבק בצורה כזאת, ואני גם מנסה ויזואליזציה, שברור שזה... כאילו, אני רואה את האפקטים מספיק חזקים שאפשר להראות להגיד, ואין פה איזה עניין של רעש, כן, רואים שזה משהו.
לא, נכון, אבל בגלל זה גם יש לך בדיוק את 4 האלה שהוא קורא להם. כאילו, אני פשוט מסתכלת על... אני מניחה שהמדגם שלי גם א' או בפחות יציב, כי... קודם כל, אותי זה בסדר. בכלל, בתוכם לקחת מסקנות זה כבר... ואז גם אין לי מספיק מהם. שתי טרדות כאלה. טוב, אני קוראת את הסיפור ורואה משהו מאישי של אוטיזם.
שינוי גדול מאוד, יש כאילו, לא עושים את זה הרבה, אבל יש איזושהי גישה שאומרת שבמקום לעשות מקום, בחוץ של בנוסף לאחרי נסיעה גדולה,
כאילו ולהראות שלכל אחד מהם יש.
כמובן, ברור שיש שונות בין אנשים שאת לא פוגעת בצורה כזאת, אתה לוקח התקדמות של אפקט מסוים.
תודה.
Next Thursday, we're going to meet here. Thursday? Yes.
Why? What's going on Thursday? We don't have class?
Yep. I was looking for a reason. Thank you.
But we're here. We're going to have a waking seminar.
No, we don't. Thursday. You're invited.
Thursday.
לילי משהו.
לא, אבל ביום לפני ליל הסדר, תגעת?
מה אתה כאילו...
אני אוהבת, ליל הסדר, חברים, ליל הסדר התחיל.
Alright, so let's continue. Yeah. Okay, so let's summarize again what we're measuring when we're measuring EEG. The first thing we can say is that when we put two electrodes on the head and measure the EEG signal, what we're measuring from the brain is everything. And I'm gonna, so this is the same review, so now it's in the right place. Quote from this review is, any excitable membrane, whether it's a spine, dendrite, soma, axon, or axon terminal, in any type of transmembrane current, contributes to the extracellular field. The field is the superposition of all ionic processes from fast action potentials to the slowest fluctuations in glia. So, yeah, we're measuring everything, but we're not really measuring everything. First of all, of course, we don't have enough independent sources of data to know everything that's happening in the brain. So first of all, like we said, whatever we are measuring has to be aligned and synchronized in time, simply to have a strong enough overall change in the electrical field for us to measure it on the scalp. So this means, so one implication is that synaptic potentials are considered to be the largest contribution to the EEG signal, and this is also true for local potentials you measure with invasive electrodes. And this is because synaptic potentials tend to be slow enough so that when they happen more or less at the same time, they can sum up together. Okay, so we're talking about sub-threshold synaptic potentials, as opposed to action potentials, which are much stronger in terms of their absolute voltage change, but they happen very fast. So they have around less than 2 milliseconds, so that's about like 500 hertz. So it's usually too fast to effectively synchronize across a large enough part of the cortex for us to measure an increase in signal. So if all of the neurons in a small patch of visual cortex fired exactly at the same time, we would have no trouble measuring a very large spike on the scalp. Something probably bad would happen to the person if that happened, I guess. But this doesn't really happen. On the other hand, if firing rate is synchronized enough, not at a spike, not precise synchronization, meaning spiking happen at the same time, but a large population of neurons being generally firing at the same time, this can be measured because it affects high frequency power, and I hope we're going to have time to mention this at the end when we talk about spectral analysis. Of course, there's other sources which are somewhere in the middle, but we're not going to get into each one of them. So measuring, yeah.
How do you differ in the EEG plot itself between the different potentials?
What do you mean by different potentials?
You have the action potential, which like have this really short increase
Yeah.
in voltage, and then you have like a postsynaptic potential.
Right. So again, when we're measuring on the head, we're measuring on the scalp. What we're seeing is a sum over all of these kinds of activities, over all of the areas of the brain, each one of them depending on how far it is from the electrode and the orientation and the strength of activity, etc. So there's no way to, we're not differentiating between anything. We're getting one aggregate signal, and we're not saying this part of the signal comes from presynaptic potentials, this comes from spikes. No, we're just saying what we are seeing, this overall sum, is mostly derived from postsynaptic potentials, because these kind of signals have the most, are the easiest to pick up when you measure EEG, because they are slow, because they tend to sum up more easily. Yeah, you seem unsatisfied with the answer.
I'm really skeptic about this method.
So let's hold this question, and we're going to talk about what we do with EEG, and then you can ask this question again. Because what we're not doing with EEG is saying this kind of stimulus causes this type of basket cell neurons to fire more and respond with, so this is not the kind of thing we do with EEG. So of course it would be a very bad tool for this kind of questions to ask. Yeah.
So how, maybe the question is not quite related. So how to reconstruct the 3D activity?
Right. Yeah. So great question, because this is what I'm talking about now. So a metaphor that Leo and my advisor likes to use is recording neural activity from the scalp is like standing outside the stadium and trying to figure out from the noise what is happening on the field, what each of the players are doing, which is kind of difficult. But actually, it's not really the case. It's more like standing outside Wimbledon, where there's a lot of tournaments going on at the same time, and standing outside this complex, and then trying to figure out what exactly is happening to the ball in each one of the courts. Of course, it's not really possible. So in principle, what we would like to do is to be able to record something from the scalp and say exactly where it came from in the brain. I'm just going to say quickly, this whole first part, I kind of feel now that I'm going to leave you with a feeling that you can't do anything with EEG. It's really not true. Just starting with all the limitations and problems, but there's really a lot, obviously, that you can do with this. You just have to use it for asking the right questions. Ideally, we would like to know exactly what the intracranial source of each kind of activity that we measure on the scalp is. So the way to do this is, of course, to use models that try to compute the intracranial activity from what we measure on the scalp. The first step to do that would be to understand how intracranial activity is reflected on the scalp. Okay, so what kind of transformation is happening there? And this is called the forward model or forward solution, meaning we take intracranial activity and we try to solve what kind of manifestation on the scalp this activity would create. So one way to do this would be to use a very simple the spherical head model with maybe 3 different kind of conductive tissues, the brain, the skull, and the scalp, and to put a dipole in here and say, okay, this would create this specific topography electric field on the scalp. So basically, this is just saying we have our vector of measurements, M. We have M electrodes. And then there is some kind of, and then we have our sources. We have N sources inside the head. Of course, this is very simple model. And we have this kind of matrix, it's called the lead field matrix, which tells us how we transform, the transformation that we make we make from these sources to the measurement, of course, with some kind of noise term added. Okay, so we can assume this kind of simple model. Of course, we're not only assuming that the head is a very simple spherical model, but we're also assuming that there's a finite number of sources inside the head. Of course, this is arbitrary because what is the source? I mean, is it a neuron? Is it a cortical column? Is it a synapse? So, again, this is a very crude simplification. But let's say we agree on this model. Then the problem that we have now is that there is no single inverse solution to this problem. So since what we know is not this, but this, we only know what we measured on the scalp from our few dozen electrodes, and since there are obviously more than a few dozen distinct electrical sources in the brain, however you want to define them, then this the inverse solution is ill-posed because we have more variables than equations, you can say, and there is no unique solution. So basically, trying to figure out the internal source from measurement on the head is theoretically impossible, and even if it wasn't, we still have the problem that we pretended that we know we know this transformation, but we don't really know it. It's more complicated than this model that we assumed is right. So the inverse the problem is theoretically impossible to solve. However, of course, we're not okay with something being impossible, and we try to do all kinds of workarounds. So I'm really not going to get into this, but there's this whole field called source localization, where we have EEG and MEG data. Oh, I didn't say any word about MEG, so I'll do it very quickly. MEG is very similar to EEG in many ways, but what we're looking at is the magnetic field and not the electrical field. One important difference in this context is that the magnetic field is not affected by the tissues of the head. So when you record the magnetic field, when you have a dipole and you have and you record it from a different specific location, it doesn't matter that there's brain and scalp and skin on the way. It doesn't affect the signal. So there's other problems with MEG, but this is a sort of advantage that MEG has. Of course, it doesn't make this inverse problem more solvable. So basically, the idea of source localization is to impose constraints on the solution space to have a unique solution. The simplest approach is called a dipole fit. Pretty much what you do is you decide on a forward solution, you decide on a head model, and then you just say, I have only 3 dipoles in the head, and they're supposed to explain all of what I'm measuring on the scalp. And then you just do some kind of gradient descent algorithm where these dipoles are shifted around and changed orientation until you achieve the best fit to your results. This seems very crude, but I have to say, for example, yes, you have to have a small number of dipoles for this to work, to have a stable solution. It seems kind of silly, but you can use it. You can use it to really you really don't like it, okay. But here's an example from my work, for example, where I had this. I show these sustained visual stimuli to my. Do you want to maybe take a short break? To my subject, and I have this kind of sustained response. I have this transient response at the beginning, and then sort of sustained potential, and sort of offset response when the stimulus ends. And I just got this weird topography of this effect on my head, and I just use this approach to see that I can not really to say where in the brain this comes from, but just to see that there is a reasonable arrangement of brain dipoles that would create this kind of topography on the head, and I really actually got a very nice result where these onset and offset related responses were localized to this occipital source, and then the more sustained component was localized to a different dipole. So again, I'm not using this to say, yeah, so these responses come from a neural population that's located here around the calcarine sulcus, whatever. I'm just saying I can explain this kind of activity using this kind of a model. And then there's more complex solutions. So you use all kinds of constraints, like I skipped it entirely. Okay. Anyway, there's more complicated measures. Usually, in our labs, we don't really. Many labs use source localization to move from measurements on the scalp to nice representations of activation inside the brain. But if I just had to say one sentence about it, you have to be very careful about this and take it with a few large grains of salt. Because again, you're making a lot of assumptions when you're moving from the scalp to the brain with EEG and MEG signals. And basically, my bottom line is that I never saw a paper saying we localize this activity to this part of the brain, which is surprising, which is not what we expected. I think when people get unexpected results from this kind of localization techniques, they would say, okay, maybe it didn't work so well, because it's not strong enough evidence to go against something that you believe. So a lot of times you would say, yeah, and this, as you expect, this visual response was localized to the visual cortex, so it probably worked, but you wouldn't see people saying the other way around. So you have to be very careful about this kind of activity.
Where do the constraints come from?
So the constraints are like saying we want to have minimal energy of the sources across the brain, or we want to have a maximal correlation, spatial correlation, these kinds of constraints. Yeah, and they make sense, but just so these methods work pretty much, but they don't work well enough that you can really use them to make strong arguments. Okay, but I really didn't want to spend much time about this because this is not, so basically trying to make specific inferences about what's happening in specific places inside the brain is not the best way to use EEG, pretty much. So this is.
But is that how you kind of find information about the different components? Because we read about certain components that they use this in order to find like the different potential.
So I'm not exactly sure what you're referring to, but I will say that using source localization can be a very good tool if you're not using it to make specific anatomical inferences, but as a way of taking your overall signal and separating it into different components. For example, some people use it as a way to clean your data from artifacts. So you can use this kind of activity to take any activity that's localized not to outside the brain, for example, to muscle noise from the scalp muscles, and then just subtract this component. So this would be, for example, a very legitimate way to use source analysis. And yeah, you can just say I use source analysis to separate my data into components, and then I use this as like a processing step, but I don't make direct inferences about where the sources are localized to. I just use it as a way to separate my data in a similar way to, for example, independent component analysis, if you are familiar with it. You will be at some point. So independent component analysis is not based anatomically, but is based on separating your signal into different components based on statistical properties, and it's a sort of a similar approach to that. All right, but I want to move ahead because I really don't like this part. I want to talk about what you can do with EEG. All right, so let's talk about the event-related potential analysis. This is the most basic analysis method with EEG. It's been around for many decades. Basically, the idea is that the EEG signal is very noisy. Apart from very, very specific cases, whatever you do in your experiment on a single trial basis, you won't be able to see anything that is happening. So what you do is you run an experiment where you repeat the same event many times. For example, show a stimulus, and then you segment your EEG data around this event, and you average. So the basic model behind this approach is that we assume that whenever something like showing a stimulus is happening, there is a specific response that is typical, that happens every time we show the stimulus, and the rest of it is noise. And when we average trials, we get to this common typical response, and we attenuate the noise. Of course, this model is, you're making a lot of assumptions here because, for example, even something very, very low level, like looking at the response to a stimulus appearing on the screen, is not really, we don't really expect the same thing to happen in the brain every single time, right? Even if it's a very low level kind of process, we expect still for there to be differences in the brain response between different manifestations of the same event, and of course there will probably be differences between subjects. So when we do this averaging analysis, what we're saying is we are going to look at whatever is the same across all of these iterations. So for example, it could be that the real response, if we had an electrode on the specific cortical column in a specific receptive field and a V1 that responds to the stimulus that we see, we would get a much sharper. Let's talk about this peak. We'd get a much sharper peak. Okay, it's a much shorter one, but its latency, let's say, is maybe not. It's not a good example because in V1 you usually get the same latency. But let's assume V1 doesn't respond so deterministically, and sometimes it can respond 70 milliseconds after the stimulus is shown, and sometimes 75, sometimes 80. Okay, so there's this difference between trials. But when we average, we take this difference and we actually smooth it, so we get this kind of maybe slower, more spread out response, which is just average across all of these events. So again, ERP assumes that the same thing is happening at the same time, but really what we're measuring is the same thing, the same thing that is common across all trials, plus and whatever we call noise is either things that are unrelated, like maybe external noise, or maybe activity from other parts of the brain that have nothing to do with processing of the stimulus, but also variability of the response itself. Okay, so if the response itself has some variability in the amplitude or in the latency or the shape, then in terms of the ERP analysis, this is considered noise. Mathematically, what we're doing when we're averaging is that we're increasing the signal-to-noise ratio by the square root of the number of the trials. There's some very simple math here, which I don't want to spend time on because really I don't have much time. But basically, if you do the math of what happens when you have a consistent signal and noise that varies across time in trials, then you measure it when you actually average across trials, then the expected value of your signal would be this common, whatever is common across those trials, and the variability would be the variance of the noise divided by the number of trials. So standard deviation is the square root of that. And maybe you'll go over the math in the lab. Yeah. That's a very good question. So no. So it depends on what we're measuring, and we're going to look at different examples of ERPs later, and some of them are recorded actually in relation to other events. like the response maybe. Yeah. Okay. So yeah, any questions about ERP analysis? Is very all you know what averaging is, so it's pretty simple. So what are ERP components? Yeah.
So what is the frequency
The frequency
of
of
EEG?
the frequency of EEG. Do you mean like the sampling frequency? Yeah. So the sampling frequency, it really pretty much it's only limited by your equipment. Usually, sampling frequency is around 1,000 hertz. Usually, it's not really, it doesn't do anything to measure with higher frequency. If you do basic EEG, a basic ERP analysis, ERPs are usually slow, so even like 255 hertz is good enough. But in some cases, some people even go to like around 15 or 30,000 hertz if they have reason to. But you're not limited in terms of the physical signal. You're not limited. You're just limited in terms of how fast you can, your recording equipment can go, and how fast you can write to your hard drive or whatever.
You also, you don't want the sampling to be, the frequency to be very high. Just a lot of unnecessary data.
Yeah, exactly. So if the sampling frequency, right, determines the highest frequency that you can record. Actually, yeah, it's not in the presentation, but I should say, EEG analysis is usually, or the EEG signal, really, you can't really find much there when you go maybe about 150 hertz. Okay, I work with high frequency responses, and really above 150 hertz, it really becomes too noisy to record anything that's coming from the brain. Okay, so if you record local field potential with an invasive electrode inside the head, you can measure spikes that have this 500 hertz frequency, but on the scalp, really anything above 100, 150 hertz is too low to be recorded. Yeah. All right, but if you're doing just regular you're not looking at high frequency, but just these average ERP responses, which are usually happen in the frequency range of maybe up to 30 hertz. So you don't really need high sampling rates. Okay, so what are ERP components? So amazingly, even though it's really hard for anything apparently to come out from the brain and be recorded on the scalp, there are many things which are consistent enough to be for us to be able to identify specific cognitive or sensory events with stereotypical measurements of the ERP. Yeah, and you can maybe generally divide them into roughly exogenous and endogenous components, where the first would be related to most sensory events, to external events, and they happen, whenever you present a visual stimulus, you have this response, or an auditory stimulus. And the other kind would be more related to cognitive processes. For example, for example, making an error and getting a feedback would be associated with a specific ERP component. So this would be contingent on your cognitive state and not on the external stimulus. But this is just a very rough categorization. So I want to go through a few examples of these ERP components. So the first one is like the most, I think the best example of the first kind of ERP component. It's called the, actually there's a few of them here. So these are responses to auditory signals. So when you measure EEG and while your subject is hearing tones, auditory tones, you have a series of expected stereotypical responses. The first of them would actually, there's a series, I don't know if you learned about this, but there's this series of steps of processing whenever you get the signal from the ear. The first station after the inner ear is the brainstem, and then you go up eventually to the auditory cortex eventually. And each one of these steps produces a typical change in the electrical field. So actually, you can actually record even the responses generated by the brainstem. It's called the brainstem, the auditory evoked brainstem auditory evoked potential. Yeah, and these are so this is what is shown here. So these are very, very, very small amplitude responses, and because they're small, you need to average across a very large number of trials, maybe I think hundreds of trials, to record them. But once you do, you have this very stereotypical series of deflections, which happen really in the first 5 milliseconds of the after hearing the stimulus. And then you have these later in time, you have these more larger deflections that are more easily recorded with less trials that come from the auditory cortex. So yeah.
The small amplitude is because it's far from the electrodes?
So there's a few reasons. Yeah, first of all, it's far from the electrodes. Second of all, it's, unlike these neurons are less oriented in a optimal way. So there's not. So here you have all these auditory neurons that are oriented in the same direction along the cortical surface. And I don't know, maybe there's maybe less neurons here than here. I'm not sure, but there's all kinds of different, all of the different factors that we talked about. Maybe synchrony between the neurons. I don't know. All these factors work together. But I think the nice thing to see here is that even though the signal that we measure here really has a very, very rough spatial resolution, we can actually, there's this kind of a connection between the spatial and temporal dimension that happens here simply because we have this very typical and well-known chain of events that allows us to link latency in time to anatomical source. Okay, whatever happens in response to the stimulus in the first 50 milliseconds, we know it doesn't come from the auditory cortex. Okay, simply because it takes more time for the signal to arrive to the input to arrive to the auditory cortex. Okay, I'm going to jump over this. There are also similar very stereotypical ERP responses to visual stimulation. Something interesting that happens because of the way the visual cortex is folded around the calcarine sulcus is that you learned a bit of you had one semester of anatomy, right? So yeah, so you know that the visual cortex is folded around the cortical sulcus in a way that the input from the upper and lower visual fields is routed to the dorsal and ventral banks of the calcarine sulcus. And what actually happens is that these cortical neurons of V1 that respond to input from the upper and lower visual field are oriented in opposite ways in relation to each other. Okay, so what happens when you show a stimulus in the upper and lower visual field is that your very early visual response called C1, which comes mostly from V1, has a latency of about 70 or 80 milliseconds, actually has this polarity inversion. So when you show a stimulus on the upper visual field, you have a negative response, and when you show it on the lower visual field, you have a positive response. And this is just because the same response happens in the brain, but the dipole that I was talking about in the beginning of the lesson is oriented in an opposite direction in relative to your electrode. Okay, and I actually use this. So I said it's hard to take activity from the scalp and localize it in the brain, but you can use these kinds of manipulations or tricks to be able to do this. For example, I have this. So, as I said before, I showed my subjects visual stimuli for long durations, and I was really interested in this kind of sustained response. Okay, nobody really looked before at this kind of sustained visual response. So I was asking, does this sustained potential come from V1? And the reasoning was that if it comes from V1, it should show the same polarity inversion as the very early component. So in one of my experiments, I showed stimuli on the upper and lower visual field, and what I saw is that while the initial C1 component does exactly this inversion, the sustained response really doesn't do it. So it doesn't come from the same sources that are oriented in the same way in V1. Okay, so this is kind of a way to make an anatomical inference without going through source localization and this kind of inference inverse solution. Yeah, so okay, I'm going to skip a lot of this. I have until 1:45, right? 10 minutes, okay. So I'm going to skip a lot. Yeah, I'm going to skip the N170. I'm going to show you two examples of of how ERP research is done in my lab. So another example, there are many. There's like a whole long list of these components. Some of them are related to sensory input. Some of them are related to more cognitive processes. This is an example of a more cognitive event. The way usually you do research with ERP is that there's two different stages. One stage is to characterize an ERP component, to say, okay, whenever this happens, I get this response, and is it affected by attention? Is it affected by stimulus size? We try to characterize as much as possible what kind of event this response corresponds to. And then once this knowledge is established enough about the component, you can actually use it in cognitive research to make inferences about cognitive processes. So, for example, so the mismatch negativity is an example of a cognitive ERP, in that what you do to elicit it is that you present a series of sounds. It has a very some kind of regularity, like it can be the same sound, the same tone, and then infrequently you break the rule. So maybe you present a different kind of tone, a different frequency, and you do this while the subject is not specifically attending to these sounds. Maybe he's watching a movie or whatever, and you compare the response to the regular versus the infrequent sound, and you have this negativity that happens for the infrequent sounds. Okay, so if you take the infrequent minus the frequent, you have this kind of negative difference here. Yeah. So this is the difference way, but this is called the mismatch negativity, and it can also, it happens in a lot of different contexts. It doesn't have to be specific differences in sound. It could be also more abstract features like high-low sound versus low-high. Okay, it generalizes to a lot of different contexts. It also correlates with the degree of deviance. So if you expect a sound coming from straight ahead, and then it comes from the side, the mismatch negativity would be stronger the farther it is from where you're expecting it. Yeah. I think so. Actually, I'm not sure. I'm not an expert on MMN, but I think it's on the auditory. I'm sure you have some kind of other other.
I think there's a really good sound kind of therapy for
Yeah.
like semantic ones.
So it's actually a point. It's actually a good point about what is a component. So you have a similar thing happening when you have a syntactic surprise. You have a sentence, then you have a word that doesn't belong to the sentence. But it's not exactly the same thing because maybe it's a different latency. It's a bit it looks a bit different. But is it the same thing, or is it a matter? Is it because it's a different just like a difference on a scale, or is it a completely different phenomenon? It's hard to say because ERP components are not really well-defined entities, right? You don't define the MMN by saying it's exactly from 300 to 375 milliseconds. It goes up to 3 microvolts. It doesn't. It's more fuzzing than that. So it's hard to say where one ERP stops and another starts, but it's just a rough characterization. But the thing is that you can use this once you know what is. Yeah. Oh.
Hi.
Yeah.
If you do such a oddball paradigm where you're subjecting a deviant tone, for example, wouldn't the expectancy of that deviant tone become more expected? I mean, you've done the experiment for maybe 20 trials and you expect a boot to come.
Yeah.
So wouldn't averaging that hide the fact that you're getting more and more used to the deviant tone?
Well, okay, so it's a good question because the thing is that one of the things that characterizes the MMN is that it's a very low-level response. It doesn't really matter. Like if I tell you in advance, I think I'm not an expert in MMN, but I think if I tell you in advance you're going to hear this kind of tone and this is going to be the infrequent tone, it's not going to really affect the response rate. It's a very early response, or not very. It's the earliest response that's correlated with this sort of more cognitive processing than strict bottom-up sensory processing. So yeah, it's considered a marker of automatic detection of change. Okay, and yeah, I mean, the bottom line is that it doesn't, it works. So you do this for a really long time and you see this difference. Yeah, it's not always expected. It's not always three frequent and then one infrequent. Yeah. So Tamar Regel from my lab. Yeah.
Does this also happen when people are really trying to focus on the stimulus itself?
I'm not sure. I know the MMN paradigm specifies that you're not supposed to pay attention to the stimulus, but I don't know enough about the MMN to say how exactly it's affected by attention. Anyway, Tamar Regel from my lab wanted to use MMN to ask a cognitive question, or a neurocognitive question, which is, we know that pitch, like in the example that I gave, the pitch, the height of the sound frequency, is processed automatically in the brain, meaning that if you have this deviation, then you have a response saying that the brain detected this, that there was a difference. But is the same correct for not pitch but pitch chroma? So pitch chroma, for anyone not familiar with it, is the place of the pitch on the periodic scale. So these sounds. So this would be higher than this, and this would be higher than this. But both this and this is a C note, and this is a G note, right? In Hebrew, do and sol. So this would be the chroma. So the question is, and if you're musically trained enough, you can easily distinguish between these chromas, especially if you hear just different manifestations, just C, 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 and then a G. You should be, at least if you're trained enough, you know how to recognize this difference. So she asked, is chroma also processed automatically by the brain? So in the first control experiment, was a standard MMN experiment. So there's a series of sounds, and then there's a deviant sound, and then the deviant sound produces a different, more negative response. Of course, you're not comparing the deviant sound to the frequent sound because there are also different sounds. so it's also a physical difference. So you're actually comparing to the same sound, but in a context where it's not deviant. So all of these sounds are different, so this sound is not infrequent, okay? There's nothing special about it. So it doesn't elicit the MMN. So comparing this and this, you have this difference, which is the MMN. Okay, and very quickly, so this creates this early automatic response. However, when you, if you took very highly trained musicians that were able to easily differentiate between different chromas, but once you played them this kind of paradigm where the G sound is either deviant, where all the rest are Cs, or it's not, there was no difference. So she concluded that the pitch chroma is not processed automatically like pitch height. It's processed later and requires attention. That's actually a different part of our experiment. I think I have time. I don't have time. Oh man, can I have just 5 minutes because this is like the best? Okay, I'm gonna. Okay, so this is my favorite example of EEG research from our lab of using ERPs to make cognitive inferences. So Dvora from our lab initially wanted to study the neural correlates of regret. Okay, and she had this paradigm where subjects had to choose one of two options, and they could win or lose, but before seeing what they won or lost, they saw what they would have won or lost with the other choice. Okay, so you choose the square on the right, and then she shows you, okay, if you chose the one on the left, you would have won or lost $8,000, and now you see that you won 500. Okay, so and they are also independent of each other. So she used a well-known ERP component called the feedback-related negativity, which is known to be more negative, to vary with feedback that you get that has significance to you. So the more disappointing the feedback is, the more negative the deflection is, the response is. Okay, it's easy to remember. Yeah, and this is a very highly established effect. So we know we can count on it. And then she runs this kind of experiment, and what she expects to find. So again, you choose one. You first see what's happened in the one that you didn't pick, and then you see what happened in the one you did pick. And she expected to find that, intuitively we say, we know that these are independent, right? She tells them, but we expect that if we lost, then we would be even more disappointed if the other choice we would have won, right? This is how people work. Okay, it's worse that we would have won if we chose the other one than if both of them we would have lost, right? But what she got was unexpectedly that the FRN was actually, it went in the other way. So according to the EEG results, it seemed like they were more disappointed when they lost, if the other choice was also a loss. Okay, which was weird. And at first she thought, something is not working, my experiment is not working, my PhD, what am I doing with my life? This is all going wrong. The literature, I'm not going to go against all the literature. What's going on? But eventually, to cut a really long story very short, this led her on a path of actually discovering a new cognitive phenomenon, or a variation. It's not completely new, but a variation of cognitive phenomenon, which she called the alternative Omen effect. And again, to go straight to the bottom line, what she discovered that actually happened is that people perceived an illusory correlation between the two results. So when they have a negative result here, so when what they didn't pick was a loss, they expected the other one, the one they chose, to be a win, and vice versa, even though these were actually independent. Okay, so if you see a loss and you expect for what you chose to be a win, then you're actually even more disappointed when it's a loss. If it was a loss, so if the other one was a win, then you thought, okay, so I'm probably going to lose. And then when you lose, you're not as disappointed. So this goes the other way around. And she actually even chose this. She actually even tested it when it's very clear that there's there's a complete independence when you have to pick beads out of two separate bags, and there's no secret tunnel between the bags, and nobody is trying to fool you with the computer code, and still people somehow more expected that when they lose here, they're going to win here, and vice versa. And interestingly, this didn't happen if they didn't care about the outcome of the result, if they didn't win anything or lose anything. So again, this is my bottom line is that this is an interesting cognitive, behavioral actually phenomenon that was exposed by having this this window into the inner working of the brain by looking at ERPs. And I think I'm going to stop here. I'm going to talk about second analysis next time. So thank you very much. זה הטיה של הגמבל פלסי, שנמצאת יותר מוקדם כאן. לדעתי זה מלפני. יש הרבה מחקר על כן. כן, זה וריאציה. יש הבדלים. גמבל פלסי נחשב דבר שנדרש, כאילו צריכה קודם לבסס איזשהו רצף, ואז לפחות שתהיה סיימה הרצף הזה. זאת אומרת, אם את כאילו זורקת לסל פעם אחת ומחטיאה, אז הגמבל פלסי לא אומר שהפעם באה תפעה לקלוע. כן, את צריכה... כן, יש עוד איזה משהו קטן שהוא, אני לא זוכר, למה זה שונה, אבל כן. לגבי האפקט של האכזב, כאילו, אני רוצה להבין מה אתה שואל. אם נסתר במקור למדוד אכזבה, דרך הקורלט הידוע של זה, שזה ה-feedback related negativity, ידוע שזה קורלציה עם feedback שהוא יותר או פחות משמח. אני שואל האם יש קורלטים אחרים שלו?
תופעה חדשה.
היא מצאה תופעה חדשה שלא קשורה לאכזבה, שהיא קשורה לתפיסה של קורלציות שלא באמת קיימות בין תוצאות של שני אירועים.
הדבר האחרון לקחת זה נמדד באמצעות
התנהגותית. ה-EEG הוביל אותה לתופעה התנהגותית חדשה. בגדול התהליך היה שהיא עשתה את הפרדיגמה הזאת של לבחור אופציה משתוך שתי אופציות, לראות קודם כל מה קורה באופציה השנייה, ואז לחפש מה קורה באופציה שכן בחרה, ואז באמת לזכות ולהפסיד כמה אגורות. והיא קיבלה תוצאות שהיא לא ציפתה ב-EEG, שהפסיקה מהן שבאמת המצב הקוגניטיבי, כאילו שכמה הם יהיו מופתעים לרעה או לטובה בכל מצב, לא הלך לפי איך שהיא. היא ניסה למצוא סיבה לזה. והסיבה הזו בסופו של דבר היא התגלתה כמבוססת על הטיה קוגניטיבית שהיא קשורה מאוד דומה למה שנקרא Gambler's fallacy. ה-Gambler's fallacy זה שאם אתה זרקת קובייה והפסדת מלא פעמים, ככל שאתה תפסיד יותר פעמים, אתה תחשוב שהפעם הבאה יש לך סיכוי יותר גבוה להצליח, למרות שזה בלתי תלוי. או כדורסל, שמישהו, כאילו יש תופעה הפוכה של היד החמה, אם מישהו מצליח מלא פעמים, אז יש יותר ספק, גם אם זה בלתי תלוי, יש כל מיני הטיות קוגניטיביות כאלה. אז זה וריאציה על זה, הרעיון זה שאם אתה, יש שתי שקיות עם גולות אדומות וירוקות, ואתה אמור לשלוף, אתה שולף בלי לראות, ואם הוצאת ירוקה, אז זכית, ואדומה טעית, אוקיי? עכשיו אתה מכניס שתי ידיים ושולף. ועכשיו אתה בוחר איזה יד תחשב, כן? יד ימין תחשב. ואז אתה קודם כל מסתכל מהיד שלך ביד שמאל. עכשיו ביד שמאל יצא לך אדום. כן? זאת אומרת, אוקיי, אם הייתי בוחר את זה, הייתי מפסיד. כן? עכשיו היא שואלת אותך, האם אתה חושב תנצח או תפסיד, אחת מה שיש לך ביד השנייה. עכשיו יותר סביר שתגיד שניצחת בהינתן שפה אחד לא. למרות שיצרה ניסוי שבו האי-תלות היא מאוד מוחשית, כן? זה לא כמו תוכנת מחשב שהיא אומרת שזה תלוי, אבל אולי היא עובדת עליך, ופה זה ממש ברור. סבבה, חוץ כמו שכדי ציפיתי, כאילו, מה זה ציפיתי? כן, כאילו, בסוף הגעתי לפחות לסוף של ה... מה? כן. כן, לא, זהו, זה היה קצת כזה כל הזמן. כזה הרגשתי כאילו, צריך לעוד. כן. כן, זהו, זה מה שחשוב לי להספיק אליי קצת. אז השיעור הבא הוא לא מוכן, אבל הנושאים